Welcome to the Sage Science tutorial for the HLS-CATCH application. We will go through the entire HLS-CATCH process for users to follow to purify and analyze the intact BRCA1 gene from human or mouse blood samples using targeted Cas9 restriction digests. BRCA1 is an 88 kb gene that is located on chromosome 17. The mouse analog is located on chromosome 11 and is 63 kb in length. The genes will be located within a 200 kb catch target shown here in a standard Illumina sequencing run. The BRCA1 gene is found within this genomic region. In this instance, over 250x sequence coverage was achieved from one lane of catch. The HLS-CATCH process is undertaken in 3 stages. A cell suspension is loaded into a sample well. An SDS lysis solution is loaded into a reagent well directly behind the sample well. Using electrophoresis, cellular proteins are solubilized and removed. Intact genomic DNA remains behind, embedded on the surface of the Agarose well. Next, the Cas9 guide-RNA mix is added to the sample well. After a 30-minute incubation, the Cas9 complex performs a double-stranded cut at the flanks of the target region. In the third phase, an electrophoretic protocol is applied that will separate the 200 kb fragments and collect them in an elution well, from where they can be collected for analysis. Non-target genomic DNA remains bound to the agarose in the sample well. Prior to starting an HLS-CATCH run, some supplies and reagents must be procured. Since lead times may vary depending on supplier, users should plan ahead at least a week in advance. Note that guide-RNA and qPCR reagents can require custom manufacture. From Sage Science, you will need a cassette kit and a cell suspension kit. For ordering information for custom reagents, refer to the Sage HLS support page and view or download the PDF sourcing guide for BRCA1 enrichment. We have validated Cas9 enzymes for CATCH from 3 suppliers. You can find a table of these suppliers in the sourcing guide, along with a direct hyperlink to the purchasing web pages. Note that Cas9 is provided in different minimum amounts and concentrations from the different suppliers. Take into account the number of catch samples you plan to run and the availability in your region. For the purposes of this demo, 2-part guide-RNAs from IDT should be used. For general use, alternate suppliers can be used as well. 2-part guide-RNAs include custom oligo CRISPR-RNAs that target the flanking regions and standard sequencing transactivating CRISPR-RNAs called tracrRNAs. Duplex buffer must also be purchased. Again, refer to the sourcing guide. The crRNA sequence from human or mouse BRCA1 targets can be copied and pasted into the IDT ordering page using the link provided. When ordering, an identifying label for the guide must be provided. Use a label listed in the table or one that you prefer. Keep the default minimum ordering scale and paste in the sequence. Press Continue, and on the next page, press Add to Order to place it in the ordering cart. Press the back button to return to the ordering page and paste in the second sequence. After you add the second guide sequence, select the minimum order amount of tracrRNA and then scroll down the page to order the minimum order amount of duplex buffer. Note that IDT Cas9 enzyme may also be purchased on this page. Press Add to Order again and review the items in the shopping cart before purchasing. When pipetting high molecular weight molecules, it is essential to use wide-bore pipette tips to avoid fragmenting target DNA, and wide-bore tips can also be useful for pipetting white blood cells. Quantitative PCR should be used to determine the amount of enrichment of the CATCH target. Using TACMAN probes is highly recommended. Stock TACMAN assays for both human and mouse BRCA1 are available. Stock probes for RNase P detection should be used as a control. The catalog numbers for these products are provided in the sourcing guide. Should you decide to use CyberGreen qPCR, primer sequences are provided in the sourcing guide as well. Finally, before starting a run, blood samples must be obtained. But note that lymphoblastoid cell lines may also be used. Blood samples should be fresh to minimize degraded DNA. Using samples that have been collected and stored for more than 5 days is not recommended. Also, for best results, blood should be stored with ACD anticoagulant. It is important to use Sage Sciences cell suspension kit to prepare white blood cells for CATCH. Efforts should be made to prepare the suspension as near as possible to the timing of the CATCH run. For instructions, refer to the Mammalian White Blood Cell Suspension Guide posted on the Sage HLS support page or the quick guide that is included with the kit. To quantify cells for CATCH, we highly recommend using qubit fluorometry. 6 to 8 micrograms of DNA equivalency is optimal for a single run. Higher cellular input will result in lower enrichment. The HLS-CATCH run will require about 9 hours to complete, with less than an hour needed to prepare the Cas9 mixture and conduct various gel cassette procedures, including preparations and reagent transfers. However, the Cas9 enzyme mixture may also be prepared a day ahead of time and stored at 4 degrees C. At Sage, we prefer to make the mixture during the 3-hour extraction stage of the HLS-CATCH run and then place it on ice until needed. Also, the elution stage can proceed overnight and the eluted targets may be collected the following morning. A few steps must be undertaken prior to initiating a run on the Sage HLS instrument. The preparation of the Cas9 enzyme mixture requires 2 steps. Refer to the HLS-CATCH workflow guide for detailed instructions. First, anneal the CRISPR-RNA and tracrRNA guides. Use the following table to prepare 1 sample or scale accordingly. To 15.4 microliters of IDT duplex buffer, add 2 microliters of each of the flanking CRISPR-RNAs. Mix and spin. Then add 2.6 microliters of the tracrRNA and mix and spin. With a thermocycler, heat the mixture to 95 degrees C for 5 minutes. Remove the mix from the heat and allow to cool on the bench top for 5 minutes. Centrifuge for 30 to 60 seconds to collect any condensation. In the second step, assemble the guide-RNA and Cas9 enzyme mix. Use this table to prepare one sample or scale accordingly. To 10 microliters of the 4X enzyme buffer F provided with the cassette kit, add the entire 22 microliters of the annealed guide-RNAs. If using the IDT or aldeveron Cas9 enzyme, also add 5.4 microliters of nuclease-free water. Mix and spin. If using any Cas9 enzyme, add 8 microliters of the enzyme. If using the IDT or aldeveron Cas9 enzyme, add 2.6 microliters. Mix by pipetting up and down, and then incubate at 37 degrees C in a thermocycler for 10 minutes. Next, dilute this 1:1 with HLS enzyme buffer C to a total single load volume of 80 microliters. Mix by pipetting up and down and then store on ice until using. Again, this may also be done the day before and stored at 4 degrees C. We find it convenient to do these steps during the 3-hour extraction stage at the beginning of the HLS-CATCH Run. Most of the remaining operations involve manipulations of the Agarose gel cassette, including reagent transfers and sealing with adhesive tape. It is important to follow best practices. Remove the Agarose cassette from the package. Inspect the cassette and packaging for buffer leakage. Small amounts of leakage, 1 ml or less, are not unusual. In addition to the top adhesive seal, there are small seals on the bottom of the cassette below the gel columns. Inspect these for leaks. If they are leaking or if there's a large amount of leaked buffer, contact customer support for a replacement. If there is a small bubble under the seal, that is okay. Look for obvious leakage or dried buffer residue. Here is an example. There may be bubbles in the elution channels. If so, it is important that they be cleared. If the bubbles are not removed, they can block electroelution and affect recovery of target collection. To dislodge these bubbles, hold the sealed cassette with its surface plane vertical to the ground. Keeping the cassette surface vertical, slowly rotate the sealed cassette 360 degrees while tapping the top surface of the cassette to remove all trapped air bubbles from the elution channels. This typically requires fairly vigorous tapping. Then collect all the bubbles into one end of the reservoirs. Either end is okay. This airspace will escape after the cassette is opened and topped up with buffer before a run. Place the cassettes onto the instrument nest. Care must be taken to remove the adhesive tape. It's good to have a Kim wipe at the point where the buffer chamber is first exposed, since during shipping the air pressure inside the cassette can cause some buffer to be ejected. The tape is quite sticky. It is best to remove the tape with smooth, slow, steady motions alternating between diagonal angles. Use one hand to keep the cassette steady in the instrument nest. Once the tape is halfway removed, switch the hand positions to hold the upper side of the cassette in place. It is important to use slow, steady motions. In areas around the buffer chamber openings, the tape will be released fairly rapidly. If the user is not prepared, they can inadvertently jerk the cassette from the nest and spill buffer. After unsealing the cassette, elution modules must be completely emptied and refilled. There are 6 elution wells and a 4.5 millimeter spacing. An 8-channel pipetter with 3 tips can be used to aspirate every other well. Set the pipetter to 100 microliters to ensure complete removal of buffer from the wells. The buffer may be exchanged within the cassette through the side ports. It is important to hold the pipette vertically when inserting pipette tips into the elution modules. There are soft membranes on either side of the elution module cavity, and any contact can damage them. When emptying the modules, Insert the pipette tips slowly into the tops of the modules and remove buffer as you lower the tips to the bottom of the modules. The bottoms of the modules are hard plastic, so it is useful to gently bounce the pipette tip against the bottom of the modules to ensure that all buffer is removed. Fill the wells with 80 microliters of electrophoresis buffer supplied with the kit. After filling the pipette tips, place the tips all the way to the bottom of the modules and slowly raise the tips as you dispense the buffer. This practice is important to avoid trapping air bubbles in elution modules. After filling, the elution modules should be almost full with a slight concave meniscus. If they appear overfilled, empty the elution modules again completely and refill. If one or more of the elution modules still appears overfilled after careful emptying and refilling, the overfilled elution modules may have a punctured ultrafiltration membrane, and the affected lanes should not be used. Next, the cassette buffer levels must be filled to the top. The electrophoresis buffer reservoir is continuous all the way around the gel channel. It should be filled right up to the level of the cassette surface. The hydrophobic nature of the cassette plastic makes it difficult to completely fill the elution electrode channels with buffer. Using a P1000 pipetter, the best practice is to first add buffer until the elution electrode channels are slightly overfilled with buffer. Second, run the pipette tip along the elution electrode channels to ensure that the edges are wetted. And third, remove the excess buffer so that the level is flush with the top surface of the cassette. Once the cassette is fully prepared, the HLS-CATCH Run can be initiated. Close the lid, and on the software main screen, if necessary, press the Clear Run Data button to clear all fields from a previous run. A new run cannot be initiated unless all fields are cleared. From the Workflow File field, load the CATCH 100 to 300 KB 3-hour extraction and 3-hour separation workflow file. Place a check mark next to the samples that will be run and enter an optional sample ID if preferred. Press the Check Current button. This will engage the electrodes to the cassette and check the electrophoresis currents in the separation and elution pathways. This will take a minute or so and provide an indication that the test was successful. If a test has failed, that cassette or lane may not have been properly prepared. After the current test is complete, press Return to the main screen and press Run Workflow. This will immediately pause the instrument with instructions for the next manual steps. Users should keep the instrument paused until the steps are completed. If the run is inadvertently resumed, it can be paused with the Pause button. During the first pause, the cell suspension and lysis reagent are loaded prior to DNA extraction. Users should take extra care with pipetting, sealing, and the order of steps. SDS in the lysis reagent solution will diffuse into the gel and sample well, so this procedure is somewhat time sensitive. Because the sample well and reagent well loading must be conducted without delay, it is important to prepare the adhesive tapes by crimping one corner so the tape can be applied quickly when needed. The backing has a strong static charge after peeling away from the tape and tends to stick to lab gloves. We find that the removed backing will stick to an unfolded Kim wipe on the lab bench. Open the lid. Buffer must be removed from all reagent wells and sample wells. Since the interior of the wells are entirely composed of agarose, careful two-handed pipetting technique should be used, demonstrated here. This gives precise control of pipette tip depth in the wells and reduces the chance of creating holes in the bottom of the wells. Use a P1000 pipetter set to about 300 microliters to empty the reagent wells. Use a P200 pipetter set to about 100 microliters to empty the sample wells. It is important to avoid damaging the soft agarose walls and floor of these wells. The best way to do this is to use a repetitive dipping motion to ensure that the pipette tip is always just beneath the upper surface of the liquid column in the well. It is relatively easy to see the bottom of the well when emptying the reagent well. However, it is difficult to see the bottom of the sample well. With a little practice, the user can learn to feel when the buffer flow to the tip is blocked when the tip is contacting the agarose floor of the well. This will not create a hole in the well unless the tip is dragged laterally across the floor of the well. Load all sample wells first. Then for each lane, load lysis reagent and tape. Then repeat load, lysis, and tape for each successive lane. The sample well input volume is always 70 microliters. The well should not be completely full or overflowing. The volume of lysis reagent is approximately 200 microliter, but it is critical that the reagent well has a small concave meniscus after filling. That is, a meniscus that curves downward into the well. So it is not critical that exactly 200 microliters is used. Just fill the well as full as possible while leaving the concave meniscus. If the well is overfilled, remove a little buffer until the concave meniscus is obtained. An overfilled well will overflow onto the surface of the cassette during taping, and the tape will not adhere to the cassette surface. The overflow region will need to be cleaned with a Kim wipe, moistened with DI water or running buffer before it can be properly resealed. This will obviously slow the entire loading process and endanger the quality of the other samples. Adhesive tapes are cut to size so that the reagent and sample well ports and the elution module ports can all be covered by the tape. It is important that the separation electrode port and the elution module electrode ports are not obstructed by tape. Hold the tape by the remaining backing strip with the backing up. Looking down on the cassette, position the tape left to right and also front to back. After positioning the tape, lower the tape so that the bottom edge sticks to the surface of the cassette. Bend the tape so that it bows towards the cassette surface, and then roll the bowed tape across the ports. Immediately after this, seal the borders of the reagent and sample wells using the bottom point of a 15 mil conical centrifuge tube. It is important to focus on sealing the reagent well first and then the sample well so that the SDS lysis solution doesn't creep under the seal into the sample well. After sealing the reagent and sample well ports, outline the elution module ports with the 15 mil tube as well. Then move on to the next lane without delay. Close the lid and resume the run. DNA extraction will take 3 hours. At the end of the extraction, the instrument will again pause and provide user instructions. When complete, open the lid. At this pause, the DNA will have been extracted and the Cas9 RNA mixture must be added and injected for 2 minutes into the DNA prior to treatment. There should be evidence of foaming in the cathode chambers due to the concentration of SDS. Use a 10 mil pipette to remove about 5 mils from each cassette lane. Then replace the buffer levels with fresh buffer. The buffer can be dripped in from above so that the same pipette can be used. Next, use a P1000 pipetter to roughly top off the cassettes. This will be done more carefully later, before the final DNA separation stage. Remove the adhesive tape. Tape should always be removed carefully. The proper technique for removing the tape seals is demonstrated here. When removing the tape, it is important to avoid mixing between adjacent elution modules and also to avoid transferring potential contaminants from the sample and reagent wells to the elution modules. Both of these goals can be achieved by pulling the tape off in a diagonal motion from the tape corner closest to elution module 6. Use one steady, continuous motion so that condensation or liquid adhering to the tape does not recontact the surface of the cassette. When the tapes have been removed, remove the contents of all of the reagent wells using a P1000 pipette set at 200 microliters. Then load each reagent well with approximately 200 microliters of enzyme buffer C. The enzyme buffer will scavenge SDS, and the volume need not be exact provided it is not overflowing. Using the pipetting technique outlined earlier, empty the contents of the sample wells completely with a P200 set at 100 microliters. When removing the sample volumes, observe whether the liquid is viscous or cloudy. This could indicate SDS leakage prior to extraction. The liquid should be clear and non-viscous. Load 80 microliters of the Cas9 guide-RNA mixture into each well. Close the lid and resume the run. The Cas9 RNA mixture will inject into the genomic DNA for a period of 2 minutes. After 2 minutes, the instrument will again pause and display user instructions. For the third pause, the sample well contents will be replaced with enzyme buffer C. This will replace necessary magnesium ions that were depleted during the Cas9 RNA injection. Open the lid. Set a P200 to 100 microliters. Empty all the sample wells. This does not have to be done precisely. Then load all of the sample wells with 80 microliters of enzyme buffer C. Close the lid and resume the software. After 30 minutes of treatment, the instrument will again pause and display user instructions. For the final stage, the catch targets will be electrophoretically size selected and eluted into the elution wells. Lysis buffer must be added at this pause to solubilize the Cas9 enzyme. The cassette buffers must be topped off to maintain electrophoresis for a 4.5-hour run. Open the lid. For each cassette lane, remove the reagent well contents. Use a P1000 pipette set to 200 microliters. After all the reagent wells are emptied, load each reagent well with lysis reagent and then add the adhesive seal to the wells of that lane. The amount of lysis addition does not need to be precise, but it is important that it not overflow. Lysis reagent will prevent the adhesive from sticking. Fill the cassette bodies with additional buffer using a P1000 pipette as shown here. Close the lid and resume the run. The elution stage will take 4.5 hours, though this can be left overnight before collecting the targets. After a run is completed, we find it useful to remove the elution products using a standard multi-channel pipette. We usually transfer the products into 200 microliter PCR strip tubes or 96 well PCR plates for storage. For DNA products greater than 50 kb in length, we recommend using wide-bore 200 microliter pipette tips for collecting products. We recommend pipetting the elution module contents up and down one time before removing them from the cassette, because we have noted that the concentration of DNA in the elution modules is non-homogeneous for some samples. For DNA greater than 50 kb, always pipette as slowly as possible, even with wide-bore tips.